Well, today evolutionists claim that there is no evidence of intelligent design, but such statements are nothing more than false teachings and or lies. Our knowledge of the cell has now reached a level where we can say definitively that we know better. The microscopic realm is so full of engineering marvels that a person would truly have to be blind not to see it. And blind they are. Blinded by their worldview of atheism and philosophical naturalism. See, these evolutionists want to believe there is no God, and so they just persist in their willful ignorance. The truth is, the microscopic world is one of God's most amazing creations. As someone who's worked many, many years in biotechnology, I've spent a lot of time on a microscope. This is I don't know if you can recognize me in this picture, is a publicity photo taken when I was working at the cotton fiber production lab at Texas Tech, a research lab that was attempting to grow cotton cells in tissue culture to lengths that would allow them to be used by textile industries. So instead of growing up your cotton plant and getting your fibers that way, you would just grow up cotton cells, treat them with hormones, and theoretically get them to grow to lengths that could be used for making fabrics and stuff. What we were looking at here in this picture is the image analysis system that we had purchased. Through it, I was able to, the image that's on the microscope is being projected on that screen over to my right. And what I was able to do with this system was determine the lengths of the cells that were in these tissue cultures. And I would also have to use the microscope to determine if there was any contaminations, if there's any bacterial contamination or that kind of stuff. I'd have to dose them with some antibiotics. But after working here, I then moved to Oklahoma State and worked at a lab there, the plant transformation facility at Oklahoma State, a research lab that was set up to provide the technical support and the equipment that was necessary to create genetically modified plants. And that ended up becoming my job for some length of time afterwards. I was then brought to Bothell by a biotech company over in Bothell to be their plant transformation specialist. But suffice it to say, have a fairly significant background in cell biology and a lot of time on a microscope. Well, and what I've found is that the microbial world can be a fascinating place. It's very foreign to our experiences, and I tend to compare it to scuba diving. Scuba diving is very foreign to our normal senses, and when you enter that microbial world, you're entering something as equally as foreign to us as that. And in this place, we're able to observe the beautiful forms of these microbial organisms and the symmetry that we tend to see, like these beautiful spherical colonial organisms called a volvox, or these single-cell organisms. These are protists. You're able to watch these under a microscope, watch the elegant ways that they move and interact with their environment. I mean, truly, we are fortunate to live in these times when technologies have been developed, both microscopes and telescopes, that enable us to explore these previously unknown realms of creation. Truly, God's handiwork has been revealed to us because of these technologies like no other time in the history of mankind. Well, the ability of these microscopic single-cell organisms to move around is amazing. And it's due to their tremendous complexity, the complexity of their design, and because they were equipped with things like outboard motors. The little tail-like structure you see on this cell here is one of these outboard motors, one that's called a flagellum. We're going to talk a little bit more about the flagellum later. However, diagrams like this of the cell are very simplistic. And they're simplistic because they must be due to the enormity of content and activity found within the cell. The reality is a far cry from the simplistic diagrams that frequent textbooks. This image was produced by the Harvard Medical School and is the most detailed illustration of a human cell that's been created to date. Well, describing such high levels of complexity can be difficult because drawing an analogy for comparison from our experiences is next to impossible. Our marvels of modern technology pale in comparison to the complexity we find in the cell. Pales in comparison to what's in a single cell, much less in a multicellular organism composed of countless specialized cell types. But a cell, a regular single cell, possesses orders of magnitudes more moving parts than we will find in one of our marvels of modern technology. Even a Boeing airplane factory doesn't even come close to the complexity we find in a cell. The cell is truly more complex than an entire city, like a metropolis, like Gotham shown here. Okay, just kidding. That's not Gotham. That's from Batman. This is Seattle, if you couldn't tell. But even a city is not a fully adequate comparison, since a city cannot move from one location to another the way cells can, and a city cannot make a copy of itself within 20 minutes like bacteria can. But there are a number of interesting parallels we can draw between a cell and a city when we talk about the architecture and engineering found there. A cell has an infrastructure that you see here, a skeletal framework of girders and bridges. It also has countless machines. We call these molecular machines. Some are called enzymes. And in fact, we have factories full of machines, like the mitochondria or the chloroplast, the Golgi apparatus, factories full of machines. And we have highways that assemble and disassemble themselves, like the ones you see here, and armies of delivery vehicles carrying manufactured goods, like this little walking robot you see there, carrying a package of manufactured goods from one location in the cell to another. Off in the distance, you also see the nucleus. It has a central library of information and a way of distributing that information. What you see here is genetic material coming out of the nucleus, being delivered to one of the organelles in the cell that will read that information and use it to make a protein, which is one of the functions of DNA. Well, truly, this cell is unparalleled in its complexity and design. However, despite this realization, evolutionists today believe that the first cell developed magically, if you will, through a purely natural process of chemical evolution, that molecules became more and more complex through time somehow, and then eventually a single cell formed through what is known as either abiogenesis or an older term, spontaneous generation. Spontaneous generation was, in fact, a view held by people of old without knowledge of the cell. For example, from the time of the ancient Greeks, it was believed that some life forms arose through spontaneous generation from non-living matter. Aristotle wrote the book you see here in 350 BC, a book titled The History of Animals, where he talks about spontaneous generation as the way some organisms originate from previous living tissue, like the way maggots will appear on meat. However, this theory, spontaneous generation, was laid to rest finally by this guy, Louis Pasteur. Now, Louis Pasteur was a devout Catholic, a Christian, and he was a renowned scientist in his day, a renowned scientist. He, in fact, one of his theories, called the germ theory of diseases, was one of the most important in all of medical history. His theory was that many diseases originate from germs, which they in fact do. Louis Pasteur is the founder of both microbiology, the study of microscopic organisms, and immunology. He went on to discover and isolate several disease-causing organisms. He isolated Staphylococcus, that gives rise to staph infections, Streptococcus, that gives rise to strep throat, and Pneumococcus, that gives rise to pneumonia. He also further went on to develop vaccines for many diseases like cholera, anthrax, and rabies. But what he did to disprove spontaneous generation was an experiment that was conducted as part of a contest put on by the French Academies of Science. An experiment in which he utilized this flask that you see here. What he did was he took meat broth and put it in this flask and boiled it until it was sterile. So if you boil something long enough, you'll kill all those microbes. He boiled this flask until everything was dead, until it was sterile. But he didn't stopper the flask, meaning he bent the neck of the flask, the way you see here. It's what's called a gooseneck flask. But in the top of the neck of the flask, he didn't put a cork or a stopper. So the flask is open to the air, and with it open to the air like this, after several days, nothing grew in the meat broth. Nothing grew. Then what he did is he took the flask and he tipped it so the nutrient broth was able to get into the neck of the flask and then drained back into the flask. And after doing that, within a day, the meat broth started to become cloudy as it had been contaminated with bacteria and fungi. With this simple experiment, Louis Pasteur disproved spontaneous generation to the satisfaction of everyone, or so we thought, and also showed that microorganisms are everywhere, even in the air. Well, despite the global acclaim of Louis Pasteur's experiment, I mean, this award-winning experiment put on by the French Academies of Science. Despite the global acclaim of this experiment, evolutionists have continued to uphold the belief in spontaneous generation, or what they call abiogenesis. Charles Darwin, even himself, was advocating for spontaneous generation more than 15 years after Louis Pasteur's experiment. Louis Pasteur's experiment was in 1859. And coincidentally, the same year, Darwin published his book On the Origin of Species. But here we are in 1871, Darwin said this in a letter that he wrote. It is often said that all the conditions for the first production of a living organism are present, which could ever have been present. But if, and no, what a big if, his words, we could conceive in some warm little pond with all sorts of ammonia and phosphoric salts and light and heat and electricity were present, that a protein compound was chemically formed, ready to undergo still more complex changes. At the present day, such matter would be instantly devoured or absorbed, which would not have been the case before living creatures were formed. So according to Darwin, with a little bit of ammonia and phosphoric salts and some electricity, you're going to get a protein compound. I mean, good grief. The ignorance of such a statement is profound, giving our knowledge of proteins that we have today. Well, almost 20 years later, 20 years after Louis Pasteur's experiment, this guy, Ernst Haeckel, one of Darwin's most evangelical promoters, continued to advocate for the belief in spontaneous generation on purely religious grounds. He said this in 1876. If we do not accept the hypothesis of spontaneous generation, then at this one point in the history of evolution, we must have recourse to the miracle of supernatural creation. See, Haeckel continued to choose spontaneous generation, even though it had been conclusively disproven, because he did not like the alternative, belief in God. Then in 1883, 24 years after Pasteur disproved spontaneous generation, Ernst Haeckel claimed to have discovered proof that life arose spontaneously. Well, what was this proof? Well, they were primitive cells that he claimed to have discovered, that he named monerans, and he described like this. He says, these cells are not composed of any organs at all, but consist entirely of shapeless, simple, homogeneous matter. They're nothing more than a shapeless, mobile little lump of mucus or slime, consisting of combinations of carbon. Well, Haeckel went on to publish more than 30 drawings of these monerans, along with 73 pages of descriptive text in a prestigious German scientific journal in 1868. He also gave them scientific names. He named one of them Proto-amoeba primitiva, because a primitive proto-amoeba must have existed before the complex amoebas we have today. But where did Haeckel see these cells? He described them as simple little lumps of mucus or slime. See, Ernst Haeckel was a professor at Jena University. And Germany, at this point in history, was the home of some of the finest microscopes available. And so a person would assume that a professor at Jena University, which houses some of the finest microscopes available in the world, would have seen these cells. But the truth is, they were pure imaginings, pure fraud. Ernst Haeckel ended up committing multiple acts of fraud to prove evolution, but I consider this to be his most egregious. See, primitive cells are a theoretical necessity for evolution, but not something that has ever been observed. But look at the extent of the details in his drawings of Monera. I mean, there is a good measure of his fraud right there to include such details. And again, 73 pages of text were published. Well, his deception is perhaps even better illustrated by the fact that he made similarly detailed but accurate drawings of other microscopic organisms like diatoms, shown here. Now, diatoms are single-cell algae that enclose themselves in a cell wall made of silica, which have these beautiful, symmetrical, geometric forms. I mean, look at these cells. That's a regular optical microscope image of diatoms. Those are single-cell organisms. Well, as I mentioned, he published on diatoms. Here are Ernst Haeckel's plates that he published in a book that he wrote. There's the title of the book there in German. It's Art Forms in Nature was the title of that book. His publications show that he was well aware of the complex and shapely forms of cells. These single-cell organisms are far from shapeless, simple, homogeneous lumps of mucus. And isn't it amazing that someone can see art forms in nature? I mean, that's the title of his book, Art Forms in Nature. That someone can see art forms out there in the biological world and not recognize that behind art forms, there's an artist. You know, biologists constantly evolutionists talk about designs in biological systems, but refuse to acknowledge that there's a designer behind designs. They talk about engineering biological systems, but refuse to acknowledge there's an engineer behind engineering, a designer behind designs, and there's for sure an artist behind art. It's so sad, though. Well, spontaneous generation theories were and are today something that has almost zero direct supporting evidence. The view has been upheld from the beginning for purely religious, philosophical reasons. Franklin Harold, shown here, is a professor of microbiology at the University of Washington, and he states this unequivocally in his book, The Way of the Cell. Read this quote. Life arose here on Earth from inanimate matter by some kind of evolutionary process about 4 billion years ago. This is not a statement of demonstrable fact, a fact that we can demonstrate, but an assumption almost universally shared by specialists as well as scientists in general. It's not supported by any direct evidence, nor is it likely to be, but it is consistent with the evidence we do have. But, ladies and gentlemen, the only evidence is that life exists. And because there is no God, it had to have come about through purely natural processes, according to your evolutionist. Klaus Doss is the director of the Institute for Biochemistry at Johannes Gutenberg University in West Germany. And he is considered one of the foremost experts in the area of molecular evolution, meaning the evolution of molecules, because molecules had to get from simple to more complex, and eventually have a cell, right? Well, he said this: more than 30 years of experimentation on the origin of life in the fields of chemical and molecular evolution have led to a better perception of the immensity of the problem of the origin of life on Earth, rather than to its solution. At present, all discussions on principal theories and experiments in the field either end in stalemate or in a confession of ignorance. George Wald was awarded the 1967 Nobel Prize in Medicine for discoveries concerning the visual processes in the eye. He openly admitted to a belief in the, quote, impossible that life arose spontaneously because he could not accept supernatural creation. In this article that I quoted, Wald went on to write that the hero of the origin of life was time. Since life had billions of years to develop, what we would consider impossible is, quote, virtually certain. And that, quote, time performs the miracles, says George Wald. He furthermore expressed the view that it was just a matter of time before RNA, DNA, and proteins would be created in a laboratory by simulating ancient Earth conditions. Wald's expectations, however, were never met by scientists despite an enormity of effort. Then, interestingly, after a lifetime of analyzing the molecular structures of living organisms, Wald had a change of heart and became a believer in creation, at least in intelligent design. In a 1984 article, he explained that, quote, with some shock to my scientific sensibilities, he concluded that a magnificent mind had created the universe and life. Interesting. I found it interesting that how many scientists I've heard of that ended up becoming believers after they retired. And I have to wonder if they weren't believers before. But today, in our academic and scientific, mindset that we have today, you cannot get published if you express even disbelief in Darwinism, much less belief in creation. You cannot get published. And so, who knows, George Wald may have been a believer before, but kept that to himself until he was safe. Well, when looking at a cell, one of the questions we want to address is there evidence of design? Secular scientists claim there is not, but they refer to features in biological systems all the time as designs. And they frequently copy biological designs in a practice called biomimicry. We're always studying God's creations and using those studies to advance our own technologies, either develop technologies or improve our own technologies in what's called biomimicry. We wouldn't be looking for designs out there in nature if they didn't exist. But we are. We're actively looking for designs and we implement them in our own technologies. Lots of examples of that. Well, the new powerful microscopes that we have today are one way of realizing design. Let me back up one second because I forgot to mention something. I will argue that for some things, you could just look at them and tell their design. For example, Mount Rushmore is seen here. I think if a person went to Mount Rushmore and they looked up at those faces on that mountain, they would be pretty sure those weren't the result of weathering. That someone had got up there and then actually chiseled those faces. But when you find an arrowhead, you can usually tell the same thing. You find an arrowhead on the ground and you can usually tell that it's not a normal eroded rock, that it has been tooled by the hands of an intelligent mind. Well, I will argue that the cell is similar, that even just looking at the cell can tell you that it's designed. And for this, these powerful microscopes that we have are helpful. Now, one of the most advanced microscopes that we have is shown here. This is an electron microscope. There's different kinds. There's a transmission electron microscope and there's a scanning electron microscope. This is a scanning electron microscope which can magnify things up to 100,000 power. 100,000 power. Well, at such magnifications, we can see some exquisite detail in cells like those diatoms. Let me remind you what the diatoms are. Here's a picture of a diatom that I showed you before. Now let's look at what those diatoms look like under the scanning electron microscope. It's a diatom. Those are single cells, two single cells sitting right next to each other. Now I say that if you found one of these, if it was the size of a shoe or a football or something laying on the ground out there somewhere, you would assume this was something that had been made by man. That we had made that it's for something, some sport or something. That just looking at these things tells you that they're designed. Or look at this one. Same thing. I think if you found one of these laying somewhere, you would just assume that what you were looking at is the result of design, of engineering. I want to show you one more from the scanning electron microscope. But before I do that, I want to show you what the organism looks like under an optical microscope. This is a coccolithophore. It's a type of algae, and it coats itself with protective plates made out of calcium carbonate. If you watch closely in the image, where right then the technician was focusing, you could see those protective plates that this single-cell organism is coating itself with. Okay, I just wanted to show you what it looked like under an optical microscope so you could better appreciate this, what it looks like under a scanning electron microscope. The structural design features of those protective plates covering these cells should alone be enough to convince an observer of the reality of intelligent design. But a worldview can blind a person to even the most obvious of evidence. But look at those things. Now, these are one of the sad things about the microscopes we have today, like these electron microscopes, is we can't look at living cells under them. So these are dead cells. They coat them with metal and then they bombard them with electrons. But I mean, even on living, you can see the structural design